We studied that one of the defining features of life is cell. Cell is the fundamental unit of life. Acellular means without cell. So how can there be a living organism without the cell? Well, there is, and now we know that there are quite a few. Let's begin at the beginning. In the year 1876, Adolf Meyer studied tobacco mosaic disease, which could be transmitted from one plant to another. He thought that it was transmitted by a bacterium. In 1892, Ivanovsky studied tobacco mosaic. He concluded it to be caused by a bacterial toxin. How did he came to this conclusion? He took the juice of the leaves of infected tobacco. Now these juices were expected to contain the bacterium which was causing the infection. So he filtered this juice with bacterial filters. Bacterial filters are supposed to trap the bacteria. Now the juice which comes out of the filter will not contain the bacteria and so will not cause the infection. But when this filtered juice was sprayed on a healthy plant, it still could cause the disease. So it was concluded that the juice or the infective agent in the juice was not the bacteria per se, it was some bacterial toxin. Continuing Ivanovsky's study, in 1898, Vigerin concluded that tobacco mosaic was caused by a living organism. But this organism was not bacterium, it was another microbe. This microbe grew in dividing cells, becoming stronger every time it infected new plants. He said the infection was due to Contagium vivum fluidum, or the infectious living fluid. He coined the term virus, meaning poison or venom. Stanley in 1935 crystallized tobacco mosaic virus TMV. He found that the molecular structure of these viruses TMV was mainly protein. He got a Nobel Prize for this. It was only later that TMV was found to contain both proteins and RNA. But this was made possible only after the discovery of electron microscope. Then the structure of viruses became clear. Now, what is the significance of crystallization of virus? Crystallization proved that these living organisms, that is the viruses, are capable of staying in an inert state where they are like non-living things. They can become active once they enter the host cell. So what are these amazing things? Viruses are not cells but particles. They are made of two components, the genetic material, that is DNA or RNA, and the protein. Remember that at a time in a virus, it has to be either DNA or RNA. Never both. DNA and RNA cannot be present together in a virus. It has to be either DNA plus protein or RNA plus protein. So the viruses are chemically nucleoproteins. When a virus infects a host cell, it is only this genetic material, that is DNA or RNA, that enters the cell. The protein code is left outside. Then the virus uses the host material to make its protein, to replicate and to form multiple copies of itself. The entire structure of viruses is known as virion. The outer protein coat is known as the capsid. Now inside is the genetic material, that is DNA or RNA. The capsid can be of different shapes and sizes. So on the basis of the shape and size of the capsid, that is the characteristic of capsid, virus classification is done. Now the protein coat or capsid are made up of many subunits. These subunits are known as capsomeres. The capsomeres are arranged as helical or polyhedral forms. On the basis of capsid characteristic, viruses are classified into two categories, helical and polyhedral. Helical viruses have capsid that forms a hollow cylindrical structure through which the genetic material passes. This is what you just saw in the structure of TMV. Rabies virus also have similar structure. Polyhedral means many-sided. So polyhedral viruses have capsid arranged in a polyhedron, usually a regular icosahedron with 20 faces. Each face is in the shape of a triangle. The spikes coming out of the protein coat are complex made of proteins and carbohydrate. Now, poliovirus is an example of such a virus. In enveloped virus, the capsid is covered by an envelope. So in this case, whether the virus is helical or polyhedral, it will appear spherical. Example, the influenza virus and the herpes simplex virus. Then there are some viruses which have a complex structure, such as the bacteriophage. It has a head which is polyhedral and a tail which is helical. On the basis of where it is causing infection, which organism is its host, viruses are also classified. Before that, you have to remember two things. One, viruses are very specific with respect to their host, and second, viruses are obligate parasites. That is, they cannot live outside their host. Now the classification. Plant viruses, they usually have single-stranded RNA inside them. Animal viruses may have single or double-stranded RNA or double-stranded DNA as the genetic material. And bacteriophage, that is the viruses infecting bacteria, they have double-stranded DNA as genetic material. Now, viroids were discovered in 1971 by Diener. These are made up of free RNA. The RNA is single-stranded and it is of low molecular weight. There is no protein coat as it was found in the viruses, hence the name viroid, virus-like but smaller. And these cause potato spindle tuber disease. You can see the potato on the right side have turned into a spindle shape. Next group of acellular organisms are prions. Prions are made of proteins only, and these proteins are abnormally folded protein. Now these are similar in size to viruses but extremely resistant to heat, chemicals and radiations. In animals, they cause bovine spongiform encephalopathy, which is commonly known as the mad cow disease in adult cattle. BSE or mad cow disease. This is a degenerative disease and it is fatal in advanced cases. It affects central nervous system. In humans, a similar variant of this mad cow disease is Creutzfeldt-Jakob disease, which is again a neurodegenerative disease which turns fatal in advanced stages. Finally, the lichens. Lichens are symbiotic associations between algae and fungi. Symbiotic associations are where both interacting organisms are mutually benefited. So lichens have two components, the phycobiont or the algal component and the mycobiont or the fungal component. The alga prepare the food, so the algae here should be a green algae or maybe a cyanobacteria. The fungi provide shelter and it helps to absorb mineral nutrients and water for its partner. So these organisms mutually benefit each other. Their association is so close that if you saw a lichen in nature, you would never imagine that they are two different organisms within them. And that is why classification of these organisms is very difficult. On the basis of structure, lichens can be fruticose or crustose. Fruticose is when thallus is bushy and crustose is when it is crust-like. Now this is why we need to know about the lichens to study them, what is their importance, how they are beneficial to us, and in what way are they harmful to us. With this, we come to the end of this session and also of the end of chapter 2. I am giving you some questions to help you assess your understanding. Try solving them once you have understood the content of this video. Happy learning. Bye-bye. Thank you.